Welcome to Unit 4, Lesson 1. Today we're going to look at an introduction to the cell. And there's quite a list of things we should be able to do by the end of today. And hopefully in the mandatory portion we're going to get up to being able to describe the difference between prokaryotic and eukaryotic cells. And then in class we're going to go over the endosymbiotic theory and the evidence that supports it, as well as looking at some common human cell types. So to begin with, what is a cell? The simplest answer is the smallest living structure. To be considered the smallest living structure, it has to be able to meet the requirements of life. And that means we're going to have to think all the way back to Unit 1 and our mnemonic about what living things are required to do. And I'm going to give you a hint and see if you can come up with the rest of the mnemonic. Starts out with responsible. Now you might have had your own. If you did, you can write it here. What's next? Hopefully you said super Grover. Aims high and really overachieves. So cells are the smallest thing capable of meeting all of these requirements. And of course, this is just a mnemonic, so what does it really represent? Okay. Responsible super Grover responds to stimuli. Grover for growth. Aims for acquires materials and energy. High for, yes, our favorite, homeostasis. Really for reproduce. Over for order or organized. And achieves for adapts or evolves over time. And cells meet all these requirements. So that leads us to what is cell theory. And basically the cell theory has three tenets. Or tenets is another word for saying principles, key ideas, or so-called truths. And the three tenets to cell theory, the three things we know to be true about cells, are that 1, cells are the most basic or smallest unit of life, as we defined above. Not that they aren't made up of smaller parts, but those parts on themselves, by themselves, cannot perform all the functions necessary to life. 2, all living things are made up of 1 or more cells. So there's no other basic structure of life that we're aware of. And 3, the real tricky one, all cells come from pre-existing cells. And we know these things to be true by observing life around us. The only problem with number 3 is, where did the first cell come from? And we're not really sure of that answer, but we know nowadays all cells come from pre-existing cells. So if we're going to study cells, it's a good idea to put in perspective how big one is. And they really do range in size from bacteria, some of the smallest cells being about 1 micrometer in size, all the way up to nerve cells, which they don't show on here, but are fit in about here, and some muscle cells, which are up to 1 meter in size. Now theoretically we should be able to see these with our eyes because we can see things that are 1 meter long, but they are so thin that we really can't see them even though they're very long. It's kind of like when you walk into a spider silk, it's just so fine you sometimes don't see it even though it may be a very long strand. And some examples of cells are the very tiny bacteria, as I said, about 1 micrometer, up to a human cell, which is just over 100 micrometers, to a frog egg, which we can see with our human eye, unaided, it's about 1 millimeter, all the way up to an ostrich egg, which is also a cell, which is very easy to see. Up until the point it's fertilized, it's a single cell. So the circle things equal cells. Okay. Now, not all of us are comfortable with the concept of micrometers to meters. So 1 micrometer is 1 times 10 to the negative 6 meters, or 1 millionth of a meter. So very, very tiny. Now if you compare it to the width of the dime, the typical cell is usually about 10 micrometers to 100 micrometers. Meaning 10 to 100 cells can fit across width of a dime. Now if we think back to DNA, that's compared to half a million double helixes, and plural for helix is actually helices, that can fit across the width. And just to remind you here, I've put a little picture of the dime. The width is here, right? Not the diameter, the width. So that tiny 1 millimeter width. Okay? So what's I find really amazing about the cell is you're going to figure out that it's like a very complex, coordinated city functioning in this tiny, tiny space. Now, there are many different cell types, but the two main divisions between cell types are prokaryotic cell versus eukaryotic cell. So we're just going to put a line here. And we're going to compare the two. Now some of you might have learned this in Bio 11. We have prokaryotic and we'll just say prokaryote. That's a noun. Prokaryotic is the adjective. versus eukaryote. And it comes from Greek. Pro means before, and karyot comes from karyon, which refers to kernel. And what they were describing as the kernel is the nucleus. So prokaryotic means cells that existed before the nucleus. And eukaryote refers eu, again Greek, referring to true. I remember you true because they rhyme. Again karyon for kernel, which was their word for nucleus, and means true nucleus. That was our original definition, prokaryotic versus eukaryotic. Prokaryotics don't have a nucleus, eukaryotics do. But we found there was a bit of a problem with this definition because there are examples of eukaryotic cells, for example, red blood cell, that don't have a nucleus. So they took the definition a little bit further, and now prokaryotic means without membrane bound nuclei, or organelles, sorry. Whereas eukaryotic means with membrane bound organelles. And that's a great definition, except for you have to know what organelles are. And these are distinct and discrete functional units within the cell. So things like mitochondria that are associated with making or transforming energy, lysosomes that are associated with digestion, they all have their specific unique functions and structures. The other way we make an analogy to these things are eukaryotes have a nice place for everything. They're organelle. They put everything in its own nice, neat little place. So these are like a tidy room, with the organelles being little shelves, cupboards, drawers where everything goes, whereas the prokaryotic is like a messy room. Everything's just lying out sort of in the center of the room with no particular place. And our examples of prokaryotic cells mostly are bacteria, and these are typically unicellular. And by unicellular, we mean they're kind of loners. They hang out on their own. They don't need others to function. In contrast, when we're talking about eukaryotics, we're talking about things like plant cells and animal cells, which are multicellular, so they usually need other cells around them to properly function. And also things like yeast, which are unicellular, meaning they don't need other cell types around them to function. Okay, so I'm just going to stop there. That's the mandatory portion. If you have any questions about that, come ask me in class. Otherwise, I'm going to pause for 10 seconds and then I'm going to resume into the optional portion in case you were away. Okay, so if you're still watching, I presume either you want to get ahead or you were away. And what we're going to talk about now is the endosymbiotic theory. This is a theory that attempts to explain how eukaryotic cells evolved from prokaryotic cells. So how eukaryotic cells in brackets with their organelles, with their membrane-bound organelles, evolved from prokaryotic cells. And it's quite a nice story. We're going to draw it sort of in panels. So in our first panel, we have a mixture of prokaryotic cells. We might have a large prokaryotic cell, and then we might have a smaller aerobic, in other words O2 using prokaryotic cell. And then we might also have a smaller photosynthetic prokaryotic cell. And in theory, all of these existed in early Earth in the same environment, presumably in the water. And the aerobic cell was functioning using oxygen to do its metabolism with, and the photosynthetic cell was functioning using CO2 and sunlight to do its metabolism with. And what we think happened is that large prokaryotic cell got hungry. So the larger cell engulfs the smaller cells. So basically, it wraps its membrane around and takes in the smaller cell. Such that that smaller cell ends up inside the larger cell. Now typically it would digest this and use it for materials and energy, but for some reason it didn't digest it, and it continued, the aerobic cell within the larger cell continued to do aerobic metabolic processes using oxygen. We think this guy then further engulfed the photosynthetic bacteria. Till it ended up having both of the smaller cells within it. Now I want you to think about what organelles would these two things become, the aerobic oxygen using cell trapped within the bigger cell versus the photosynthetic one. If you said that this trapped smaller cell became the mitochondria, you'd be correct. Remember, the mitochondria uses oxygen to break down glucose. And then if you said the photosynthetic one, which isn't in all cells, but if you said the photosynthetic one represented chloroplasts in plant cells, you'd be correct according to the theory. Okay, so this is how they think organelles started to form inside of the more simple prokaryotic cell. And the reason it's called endosymbiotic theory, endo means within or internal, and symbiont, think back to grade 10 science, or to something like alien. A symbiont is one organism that lives in or on another without harming it. And often helps it. Now it's thought these things had a symbiotic relationship that was actually mutual, in that the larger bacteria that engulfed it protected the smaller one from being destroyed, while the smaller one provided energy to the bigger cell. Okay. Now that's a really great little story, but this is a theory, not a story. So it needs to have scientific evidence. And there actually are at least 5 pieces of evidence supporting this story. So although the little Pac-Man analogy is cute, it also has evidence. So the first thing is, in terms of if we compare these supposed bacteria with mitochondria and chloroplasts, we find out that the mito and chloro, short for mitochondria and chloroplasts, are similar in shape, size, and structure to the bacteria that they supposedly derive from. Second, mito and chloroplasts are surrounded by a double membrane. So 2 lipid bilayers. The outer is thought to be from the large prokaryote engulfing while the inner is thought to be the original membrane of the engulfed bacteria. So if we go back up to that picture above, we have this double membrane for the mitochondria, the red being the inner from the original bacteria, and the black being from how this actually stretches its membrane around and engulfs it. Three. Mitochondria and chloroplasts have their own unique DNA. Called plasmids, just like bacteria have plasmids. And that can replicate and divide. So we think this genetic material is a leftover from the days when they were independent bacteria. As well, mito and chloro have their own ribosomes and can make some of their own proteins. We think this is a leftover from when they were an independent bacteria. Nowadays, most of their proteins are made the traditional way, DNA from the nucleus turning into mRNA, protein being made in the cytosol and directed through the ER to the actual organelles, mitochondria and chloroplasts. But they do have some that are made entirely within the mitochondria or the chloroplast. And finally, perhaps the most interesting is that the RNA sequence in the ribosomes are more closely related to that of bacteria than to our own ribosomes in our cytosol, suggesting that they actually came from a different origin. Okay? So that's the endosymbiotic theory and the evidence for it, and when you add up all the evidence, it's actually a pretty convincing theory, although at first it seems like a fantastic but fun story. Okay. So just to finish off our introduction to the cell here, we're going to look at what are some examples of human cell types. And to start with, there's approximately in you 100 trillion cells in your body. And just for those who like to think in exponents, this is 10 to the power of 14. And there's many, many different types, so we're just going to introduce you to a few types. But to remember, what makes different cells different? Different is that they are expressing different combinations of proteins. And this leading to different structures and functions. And we're going to learn more about the specific structures and functions as we go through the various systems, but right now we're just going to take a quick overview. And you should know that these aren't drawn to scale. But some of your major classes of cells there's going to be nerve cells, and within that there's at least 3 different types we'll discuss. You have muscle cells. We're not going to look at muscle cells because the muscle system isn't one of the systems we look at in Bio 12, but we'll mention them briefly when we talk about how cells function. Bone cells, again, skeletal system isn't something we really look at in Bio 12. Reproductive cells, we will be discussing in detail. Sperm and egg, or more fancy word, ovum or oocyte. Blood cells, we will be discussing in detail, including our red blood cells. Think about Ralph. And he is the exceptional eukaryotic cell. Because, and I'm not going to tell you right now. If you can't remember why, come ask me. Okay? And gland cells, and we'll be talking about several different types of gland cells, in particular with the digestive unit. Okay? And then as you can see here, they all have different structures, and that relates to the type of functions they're able to perform. And it's a result of expressing different types of proteins. If you have any questions, you can come see me in class. Otherwise, I hope you have a good day, and I will see you in class.